This video is about the development of a human embryo. So we start out here, and I want to make sure that you have your diagram sheets and you can follow right along with me. Feel free to cut up the sheets, put them into your notes, add things to it. Make sure you also go through your review book and add notes from your review book as Cornell notes to this. And your textbook has good diagrams and good descriptions as well. So we start off here with our oocyte. So the oocyte in the ovary has gone through follicle development and ovulation due to that spike in luteinizing hormone, and it has been released, and it is now traveling through the oviduct. So it is a secondary oocyte. It is still stuck in metaphase 2 of meiosis, and this is where fertilization occurs right here. So fertilization, the sperm have worked through that zona pellucida and fertilized the egg. It now has gone through its final stage of meiosis, released the rest of the DNA, and is a normal diploid cell. This fertilized cell is called a zygote. The zygote then continues to travel down the oviduct, and it's now going through a process called cleavage. Cleavage is mitosis. It is dividing and becoming 2 cells, and then 4 cells, and 8 cells, and so on. So this mitotic cell division, it doesn't really increase that much in size, but it's gaining in cell number. And eventually it forms a large mass of cells. That is called a morula. And then this is called a blastula. A blastula is when it starts to hollow out the center portion of it. And the blastula is also where it embeds itself in the uterine wall. These steps all together take about 1 week to occur. All right, here again are those stages: zygote, going through cleavage and forming more cells and more cells until you can see it as a hollow ball. So there's a cross section of it as a blastula. And then it's sort of like if you imagine a tennis ball and you push in with your fingers. It is doing this. This is called gastrulation. When it becomes a gastrula, it begins the differentiation process where cells start taking on different roles. So you will be able to see this is called an archenteron. This isn't labeled. That arched area. The blastopore is the first opening. So in humans, that first opening is the anus, and then this top part right here, the endoderm, when that fuses with the outer part, that's going to form the mouth. So therefore you will have you're basically a tube within your body, and that tube is your digestive system. So that's where that comes from. Now these 3 tissue layers, they're called germ layers. Each germ layer is going to take on a different job in your body. So we're going to write 3 germ layers. The outermost layer is called the ectoderm. And the ectoderm gives rise to your skin and your nervous system. So think of it, that is the most outer part, and that is what is the outer part of your body. In from that, the last one to develop is called the mesoderm. And the mesoderm is going to give rise to muscles and bones and blood vessels. And then the innermost layer, where that beginning archenteron pushed in, that is the endoderm. And that is going to make up your digestive tract, and I'll just put here other very internal organs. So things like your liver would form from there as well. All right, next slide here. I just liked how this picture showed the blastula and then how it pushed in. So this green would represent the endoderm. The mesoderm would start to form where the yellow is. Last to form, but that's where it would form. An ectoderm on the outside. This first opening is called a blastopore. And again, that in humans is going to make up part of the digestive system. For us, the first opening is the anus, and the last one, which will happen when this fuses, will be the mouth. Another view, same idea. All right, this one, I thought this was interesting because it does a good job showing the cells, but then also you've heard a lot about stem cells in research. So stem cells are undifferentiated. So you'd have to get them at the point where they are in the blastocyst. And if you remove those cells, they have not taken on a role yet. They aren't skin cells or stomach cells or anything else, so they have the potential to turn on any gene and become anything. So we call these embryonic stem cells, and eventually ectoderm will form something, mesoderm will form something, and endoderm will form something. Germline is those cells that are capable to undergo meiosis and form gametes. But these cells must be turned on, and to be turned on, first of all, remember they all have the same DNA. So transcription factors, things that bind to DNA and say you turn on now and you build that protein. Those will change within these cells. So in development, let me show this first. In development, let me finish my sentence. In development, each of these genes are going to be present in every cell, but in some places of your body, these genes will be turned off, and in other places of your body, certain genes will be turned on. That's what allows cells to become different. So differentiation just means the ability of cells to take on their role because of the genes that are being expressed within that cell. All right, so we have what are called Hox genes, and this has been studied a lot in fruit flies and in sea anemones and frogs, so and in us as well. But they're looking at specific genes, and they notice these genes are really active in the beginning stages of an embryo. And the protein that these DNA produce, so going through transcription, making RNA, going through translation at the ribosome, building a protein, the proteins that are made are transcription factors. So those transcription factors are what go and bind to another section of DNA, allowing RNA polymerase to work and transcribe it. So therefore, they are things that turn genes on. So they contain DNA sequence known as a homeobox. So there are certain homeoboxes that have been identified in multiple organisms that are really active in the beginning stages of an embryo. In many animals, the organization of the Hox genes on chromosomes is the same as the order of their expression along the anterior-posterior axis of the developing animal. So anterior head, posterior tail, they will actually notice certain genes in sequence that get turned on to develop certain traits for the head, certain traits for the middle section, certain traits for the tail. And collinearity, so in a linear sequence, one will affect the next, affect the next, affect the next. So here is a diagram that shows some different, these are homeobox genes, so these different colors represent expressions of different genes in different organisms. So the idea is you have genes present from the very earliest stages of development that control what different parts of the embryo will become, and they do that by producing transcription factors that bind to DNA and turn genes on. But if you remember earlier when we did molecular genetics, we talked about epigenetics. So I also want you to think about, you know what, if they're going to turn certain genes on, they also are going to turn other genes off. And how do they do that? Methylation. So I hope that you were saying methylation while I was waiting to say methylation. Methylation, they might have DNA methylation where they actually cover up that sequence of DNA so it doesn't get expressed. So for example, your stomach does not need to produce skeletal muscle. It has the DNA to produce skeletal muscle, but your stomach is made up of smooth muscle. So turn off the skeletal muscle genes, turn on the smooth muscle production genes. Another thing that it might methylate is histone methylation. So the way the DNA is coiled, it can actually coil it tighter so that whole areas of a chromosome will not be expressed. So this is another diagram that I just thought was really great. You do not need to know any of the terms of these genes, but it's so interesting to look at and see. There are certain genes that are being turned on at different phases, and the genes that are turned on allow differentiation of cells to form different organs and different tissues in your body. So this is simply to show activation of different genes allows activation of different parts of the embryo to become different types of cells and then different types of tissues. This is highly complex, but if this is something that interests you, this is showing how if you take out a blastocyst from a developing embryo here, and this blastocyst has the stem cells, so here are embryonic stem cells here, they could be induced. It's called embryonic induction. They could be induced to take on different roles depending on what part of their genes are turned on and what parts of their genes are turned off. So they show ectoderm and mesoderm and different layers and exactly which genes lead to the production of different tissues. Here's another one, same idea. These are different genes and it shows where they are most active and where they are not active. And this also shows the linearity. So here was the blastopore where it was not specific, and these cells could be embryonic stem cells. But here they have started, according to where they are located, according to their linearity, to do a certain job. So these are being turned on to actually, this is going to form a neural cord. One of the first things that forms in that after gastrulation is the nerve cord. So if you think back to the video that we watched, when you saw the gastrula embed in, the first thing that you saw form was the notochord, which is going to make up the spinal cord, and then the vertebrae around it. So they actually take on different jobs according to where they are in their plane. And there's some scientists that have actually tried taking out these embryos and dividing it or cutting it in certain ways. And they realize based on where it's cut and if it has this gray crescent, if it has certain material present, it will either allow embryonic induction to continue and it will become normal, or if it's cut in a different way, they've sort of lost their sense of what type of cell should I become. So ask questions away in class, make sure you read through your review book and go over all this stuff about embryos and turning genes on and others off.